Well, if you remember, within the previous videos we mentioned about the polyglycoproteins which work as the nanopumps and inhibit the passage of drugs through the blood-brain barrier, okay? And we mentioned that when they enter and pass through the blood-brain barrier, these drugs are pumped back into the bloodstream because of these polyglycoproteins. But in reality, it doesn't happen like that. You know, it's not like passing the phospholipid bilayer completely and then being pumped back. In reality, the drug is entered here into the phospholipid bilayer and coming here and then, you know, pumping back into the bloodstream. Okay, let's have a look at the amino acid sequence of the polyglycoprotein. You see here we have the hydrophobic membrane and the ATP part, which is responsible for converting the ATP into ADP and providing the energy for the polyglycoprotein. Okay, here we see a model which has been presented to see how the polyglycoprotein works. Okay, you see here the drug enters here to the phospholipid bilayer and then it will be recognized by the polyglycoprotein, okay, and then by the conversion of ATP into ADP, a sort of closing the zip, you know, the closing the zip of your jacket. How does it work? The zip is closed and it is pumped back, okay, go back into the bloodstream. So the drug enters here and the PGP pumps it back to the bloodstream. Okay, here is another model which have been presented. It says that not all the drugs are 100% hydrophobic. Okay, so there are also the hydrophilic parts related to this head. Okay, but due to the interaction of this hydrophobic part with the hydrophobic lipid membrane, to be more precise, we can say with the phospholipid bilayer here, the interaction of this part and these phospholipid bilayers somehow enters the compound, the molecule here, okay? But then in order to come back, in order to cross this layer, it has to flip, it has to rotate, okay, to put back, to exit its hydrophilic part first and then to get out of here completely. This is sort of an energy barrier that this compound, this molecule has to overcome. But once it does the flip, the PGP does the flop. You know, it rotates and it rotates it as well again. When it rotates, the PGP rotates it again and push it back into the bloodstream. So this is the flip-flop. Okay, in order to inhibit the function of the PGP proteins, there are some as you see here, some materials, some molecules, let's say anything that you can use, but we have to be careful, okay, by inhibiting. What is the meaning of inhibit? Inhibit means to stop. To stop the function of the PGP. If we use the inhibitors, we stop the function for anything. You know, we make the blood-brain barrier permeable to any kind of molecules. So the toxic molecules, the anything which is hazardous for the brain can be entered into the brain as well. So we have to be careful upon using these PGP inhibitors. Okay, one of the inhibitors which can be used in order to make the brain more permeable are the block copolymers, which are composed of the PEG and the PPG. Let's have a better look. As you see here, we have a block copolymer. What is a block copolymer? A block copolymer is a made of two parts, okay? Two polymers together. It is called the blood copolymer. The one part is the polyethylene glycol, which is the hydrophilic part, which loves the water, and the other one is the polypropylene glycol, the hydrophobic part, which loves the oil, the hydrophobic fluid. Okay, if we put a lot of these blood copolymers into these oil fluid, oil phase, oil liquid phase, you see here, because these parts like the oil, they will be out, and because these parts hate the oil, they will get together and form the core of these polymeric micelles. This table says that if we choose the blood copolymers with the right molecular weight and the right ratio between the PEG part, you know, the hydrophilic part, and the hydrophobic part, the PPG, we can build a library of different micelles with different sizes, okay? And at one optimal size, the activity of the PGP will be somehow inhibited, will be stopped, okay? Here you see the HLB and the different groups of the micelles which have been prepared to see whether the function of the PGP molecules are going to be stopped or not. But let's see what is HLB. HLB is the hydrophilic lipophilic balance. Okay, for the group 1, you see here that because the hydrophilic parts of the blood copolymers are bigger compared to the hydrophobic parts, they do not have enough energy to enter and pass through the blood-brain barrier. You can see the details here. For the group 1, they interact with the cellular membrane, but they cannot pass, okay? They do not have enough energy. But for the group 2B, group 3B and group 3A, you see that here a sort of an endocytic vesicle is formed, okay? So it means that here the hydrophobic parts have enough energy to interact with the phospholipid bilayer of the cellular membrane, and they can pass, okay? And also here you see that they pass. Why? Due to the lipophilic part, due to polypropylene glycol, okay? But here, due to polyethylene glycol, the hydrophilic part, they cannot pass. But at some optimum ratio, this micelle will somehow enter and pass the tight junctions and then block the conversion of ATP into ADP for the polyglycoproteins here, and then you see that the function of the PGP has been stopped, okay? So they act like the inhibitors. So this is one of the ways to increase the permeability into the blood-brain barrier, through the blood-brain barrier into the brain.